Welcome to the third video for the evolution unit. In this video, we will discuss the process of natural selection and other factors that are responsible for changes in the allele frequencies of a population. Natural selection is the change in an allele's frequency over time due to that particular allele's impact on the survival and reproduction of the organisms. It describes the concept that an individual that is best adapted to survive due to advantageous alleles leaves more offspring, and as a result, the next generation is enriched with these same advantageous alleles. You may have heard this described as survival of the fittest. When using that term, it is important to remember that fittest, or having the highest fitness, in this context is a measure of how much an individual influences the next generation's gene pool. Natural selection that increases the frequency of a favorable allele is called positive selection, while natural selection that decreases the frequency of a deleterious allele is called negative selection. In order for natural selection to occur, there are a few important characteristics. Individuals in the population must have variation in their heritable traits. It is this variation that is selected for or against during natural selection. Additionally, organisms produce more organisms than can survive in one environment, so there is always competition for resources for survival. The individuals with beneficial traits will compete better, thus having more offspring and influencing the frequency of alleles in the next generation. Negative selection does not always completely eliminate deleterious alleles, because often the allele is only lethal or deleterious when it is homozygous, which means natural selection would not affect heterozygotes carrying the deleterious allele. Interestingly, those deleterious alleles might not always be harmful. If this is the case, then natural selection will keep an intermediate frequency in the population through a process known as balancing selection. One example of balancing selection is through heterozygous advantage. This means that a heterozygous individual is able to survive better than either the homozygous dominant or homozygous recessive individuals. A good example of heterozygous advantage is seen in the mutant version of the hemoglobin gene. If you are homozygous recessive for the mutant version of this gene, this causes sickle cell anemia, and you have sickle cell disease, as seen here. Many individuals can die of this disease before having children, and thus the allele is not passed to the next generation. However, in certain areas of the world where malaria is prevalent in the mosquitoes that live there, individuals who are homozygous for the normal allele are more susceptible to getting malaria and dying. But heterozygous individuals do not have sickle cell anemia and are protected from malaria, and so they are selected for over either the homozygous dominant or the homozygous recessive. We can also examine natural selection's effect on the traits by graphing the occurrence of a particular trait in a population over time. This allows us to see different patterns of selection. There are three main patterns we might observe. The first is stabilizing selection, which maintains the average and acts against the extremes. The example shown here is human birth weight. Those babies with low birth weight have a low chance of survival, while those who are too large can cause complications during delivery, resulting in the death of both the mother and the infant. So the optimal birth weight has been selected for between those two extremes. Directional selection selects against one of the two extremes, so it causes a shift in the population towards one extreme or the other. An example is seen in one of the species of Darwin's finches, which live on the Galapagos Islands. The finch shown here is a medium ground finch that lives on seeds. In 1976, seeds were diverse and plentiful, but a drought in 1977 caused the seeds to become scarce. The small and medium-sized seeds were quickly eaten, and the birds had to turn towards the larger spiny seeds that are harder to crack open. The graph shows the distribution of beak sizes in birds before the drought, in purple, and those after the drought, in blue. During 1977, the birds with the larger beaks were better able to crack open the large spiny seeds. They got more food, thus had more offspring. So the generation in 1979 had a much higher frequency of larger beaks from their successful parents the year before. This shift in beak size towards one direction is an example of directional selection. The third type of natural selection we see is disruptive selection, which selects against the mean, so individuals at both extremes increase. In this example, we are looking at fly larvae that are feeding on two different types of trees, apple and hawthorn. The fruit on these trees blossoms at different times of year, so we can see one population of flies that are now apple specializing and one population that are hawthorn specializing. Before the apple trees were planted in the area, all the flies would have been under stabilizing selection for the hawthorn trees. Darwin was puzzled by features that persisted in a population but that seemed to reduce an individual's chance for survival. He did not understand why the peacock would continue to have a tail that was metabolically expensive to produce because it takes energy to make the colored pigments and serve as an advertisement for potential predators. It turns out that these traits are under a different type of selection than natural selection. They are under sexual selection. Natural selection would be acting to reduce the size and showiness of the tail, but sexual selection is acting in the opposite direction. Sexual selection is anything that will increase an individual's opportunity for reproduction. In this case, the peacock's trail is attracting the female mate. Female peahens choose their mate based on the showiness of the tail, and this selective pressure has caused the tail plumage to be maintained in the population. This is considered intersexual selection because it is happening between males and females. In another form of sexual selection, members of one sex, usually the males, compete with one another for access to the other sex, usually the females. Antler size is often under sexual selection due to their use in fighting for a mate. This form of sexual selection is called intraselection because it focuses on interactions between individuals of one sex. There are a number of other factors that will influence and change the allele frequencies of a gene pool. However, unlike natural selection, these changes are not in response to survival within the environment, and instead are random changes. Migration leads to the movement of alleles from one population to another, resulting in gene flow. A high level of gene flow between populations will homogenize them, or make them more similar to one another over time. Mutations result in new alleles, as we have discussed previously, which influence the frequency and composition of the gene pool and that natural selection will act upon. Non-random mating will also influence the allele frequencies because when individuals preferentially choose one mate over another according to their phenotype, and thus their genotype, certain phenotypes increase and others decrease. This rearranges the alleles in the gene pool, but often does not add new alleles to the pool the way migration or mutation do so. The factor we will discuss in more detail is genetic drift, which is just the random change of alleles from one generation to another due to chance. Genetic drift is the random change in allele frequencies from generation to generation, meaning that frequencies can go up or down simply by chance. The impact of genetic drift depends on population size. In small populations, shifts in the allele frequency can occur rapidly from generation to generation, but if the population is large, then shifts in the allele frequency from generation to generation are generally much smaller, typically less than 1%. This is analogous to if you flip a coin 6 times versus if you flipped it 1,000 times. While probability says you should have a 50-50 chance of heads or tails, there's a bigger chance that you won't see that 50-50 ratio in the 6 flips versus the 1,000 flips, just due to random chance. When we use the computer to model random changes in populations, we can see the effect of genetic drift on the small population, as after 20 generations each allele becomes fixed or completely disappears, but in the larger population, the allele frequency has not changed as drastically due to the limitations of chance on large populations. There are two main types of genetic drift, bottleneck effects and founder effects, both of which result in small population size. A bottleneck effect occurs when a large population is drastically reduced in size due to a random event, such as a hurricane or an earthquake. The random event kills off a majority of the population, and the small group of survivors likely do not have the same allele frequencies in the remaining gene pool as the original population. For example, this population of bugs has a certain allele frequency. There are 4 homozygous dominant red bugs, 3 heterozygous red bugs, and 5 homozygous recessive green. You can pause the video and calculate the allele frequencies for the dominant B and recessive B alleles. You should have calculated that the frequency of the dominant allele is 45.8% and that of the recessive allele is 54.2% of the population. This is close to 50-50. This population is living along a coastal shore, and when a hurricane comes through, it floods the coast, killing off a random section of the population. The remaining population now has a different allele frequency, with 3 homozygous recessive green surviving and 2 heterozygous red, giving us a drop in the B allele frequency to only 20% and a rise in the recessive B frequency to 80%. The surviving gene pool has had a change in allele frequencies, but not due to natural selection because the green allele was helping survival, but just due to random chance. The other common example of genetic drift is the founder effect. A founder effect occurs when a small population breaks away from the larger population and resettles somewhere else, i.e., they found a new population. The allele frequency of this new population may be different than the original due to random chance of which individuals happen to move. For example, if 4 of the bugs from our original population flew away and landed on an island offshore, they would start a new population there. In this new population, there are all red bugs, and only 1 of the 8 alleles, or 12.5% of the gene pool, is made of the recessive allele. This is much different than the original population. We see founders effects in human populations that tend to keep to themselves, such as the Amish or the Ashkenazi Jewish populations, which have different frequencies of certain traits compared to the general population. Changes in allele frequency within a population can eventually lead to the development of new species over many generations, if the population becomes sufficiently different than the other populations of its own species. For example, if we split a population into 2 daughter populations separated by a geographic barrier, then over time mutations will occur in one population that are different than those that occur in the other population. This creates new alleles and different allele frequencies. When you look at the population after thousands of generations, you will see that the many differences in the DNA sequence due to these mutations have accumulated. This is called molecular evolution, and after enough mutations have accumulated, population A and population B may consider different species. The concept of a species will be discussed in Chapter 22. Because of molecular evolution, the extent of genetic difference between 2 species is a function of the time that they have been genetically isolated from one another. This means that we can examine DNA sequence differences and similarities to determine the time that the 2 species have been evolutionarily separated, a concept called the molecular clock.